Today we're going to talk about, or I'm going to detail what happens in chapter 5 of my new book, The Quest for New Knowledge. And the title of this chapter is the interleukin-2 / interleukin-2 receptor structural activity relationships. And really this chapter is a story of my life in the 1980s at Dartmouth, leading up into the 1990s. It's basically the second 10 years of my tenure there. And it's really the story of all the people that came through my laboratory during that decade. And so the good news is that almost all of the people that sort of appeared on the scene, as I will relate, were really great people. And so not until I went back and really recounted it all in my memory and wrote it all down and so forth did I realize, you know, how lucky I really was with all these people. Right in around the early 80s, after we had succeeded in generating the interleukin-2 receptor assay and succeeded in analyzing, doing analytical biochemistry so that we could prove to ourselves anyway that interleukin-2 was only a single molecule. There were some naysayers at the time that said, well, you know, Kendall, in that conditioned medium you're using, you have no idea how many molecules are really responsible for your so-called T-cell growth factor activity, and which was a sort of a depressing idea, because if that was true, then we couldn't purify one molecule that had all the activity. Sort of goes without saying. And so I felt that kind of a criticism of the whole thing was really sour grapes on the part of most of these people. So anyway, we had finished a series of biochemical experiments that were primarily done by Richard Robb, who came to us from his postdoc in Sweden with Hans Wigzell. Prior to that, he had done his doctorate work with Jack Strominger at Harvard, and Rich was by this time a seasoned protein and membrane biochemist, because he had spent most of his time working on purifying molecules from the cell membrane that were active in immunological kinds of things. And he was very, very good, and he did a great job. And he went through separative biochemical procedures, taking conditioned medium that had the activity in it and reducing it down so that we could show that it's only a single protein. However, once we did all these biochemical separations and everything else, we didn't have just a tiny little bit, not any pure IL-2 to speak of to take it to the next step. And that is, we didn't have enough pure IL-2 to put it back into biological experiments to show, well, this is IL-2 does this or IL-2 does that, and so forth and so on. It was clear to me that what we really needed to do was to create a monoclonal antibody that would react with IL-2, the TCGF molecule that we could show that it was only one molecule. Now, that became a problem, because this was in the early 1980s. Monoclonal antibodies had been developed and created by two investigators, one from London and the other one from the Basel Institute in Switzerland. And one investigator from London was named as Cesar Milstein, and he had developed a way to fuse cells from two different kinds of cells so that their membranes would fuse together, and then you would have one great big cell with actually double the number of nuclei. That was called a hybridoma. George Kohler, who was a student at Basel Institute and the Basel Institute for Immunology, heard Cesar Milstein give a seminar about that, and Kohler said, well, to himself, he said, so let's fuse together a plasmacytoma, which is a malignant cell that will just grow continuously in the test tube, with spleen cells from animals that have been immunized with an antigen, and fuse those cells together, and then we can hopefully find a hybridoma that will grow continuously, but will make the antibody that we want. And of course, you have to detect that and find out what it is. They did that. They published that in 1975. Now, we were working in 1981, 82. For the most part, making monoclonal antibodies or doing that whole procedure was sort of an art form. It really hadn't been industrialized so that everybody in the whole world could do this sort of thing. And one of my friends came from the Basel Institute named by the name of Max Schreier. I told Max what we were planning to do and asked him to give advice to me and so forth on how we might go about this whole thing. And he said to me, I called him on the phone over from Switzerland, and he said to me, Kendall, you just don't have a prayer. You can never do that. Not in your little lab in Dartmouth. You're kidding. So, of course, as soon as anybody tells me, my mother learned this early on, somebody tells me I can't do it, then I really want to do it. So the problem is that for the most part, these monoclonal antibodies, and they're called monoclonal, because what you do is you isolate one hybridoma from all the other hybridomas that after you fuse them together, and then you grow up all the cells that have expanded as a consequence of that, from one cell, which we call the clone then. And then you can have antibodies from that hybridoma that bind to your particular antigen. For the most part, people have done this by immunizing mice with whole cells. And so you could get antibodies that would react to, say, sheep red blood cells, which is what Milstein and Kohler first used. But to use it to get an antibody against a single protein hadn't really been done, at least not on a very wide scale yet. And so the first order of business in order to do this sort of thing was that you had to have enough protein, pure, because you don't want to immunize your mouse with the whole gamish of proteins, because then you're starting out with, it's garbage in, garbage out. And so you want to immunize with the single protein, but you got to have enough of it to activate the mouse to make antibodies, to get plasma cells to make antibodies that will react with your particular antigen. And so that meant that we were on a TCGF production assembly line, essentially. And because the only source of the growth factor were lymphocytes, normal lymphocytes at the time, I decided that in order to get enough lymphocytes so that we could stimulate them to make the growth factor, we needed to, I went to the ENT department at Dartmouth Medical School, ear, nose, and throat, because they were doing tonsillectomies sort of on a daily basis. And from a single tonsil, you could get 1 billion lymphocytes. And 1 billion lymphocytes would give us a liter of conditioned medium, or a quart. A liter is about the size of a quart. And prior to that time, we'd been making like, 1/10 of that, like 100 mLs instead of 1,000 mLs in a liter of conditioned medium. And we thought that was a lot of conditioned medium. Well, we figured out that in order to get enough stuff to be shipping into these mice, we were going to have to get liters and liters and liters and liters of this stuff stacked in the cold room. So that's what we did. And I got one of the ENT guys who was just great. He would call me on the phone. I would run over to the hospital. It was in a different building, and put on my scrubs. And I'd go into the OR, and they'd give me a stainless steel cup, and I would hold it out, and he would plop the tonsil in. And I'd run back to the lab, and then we'd sit in the tissue culture hood, laminar airflow hood, and we'd grind it all up with scalpels and forceps and so forth and so on to make single cell suspensions. And then we would put them in culture and stimulate them with our stimuli overnight. And we would then get a lot of T cell growth factor by the next day. So we started banking all this stuff in the cold room. And then Rich took all that conditioned medium and he passed it through a filter to concentrate it down at 1/10 or 1/100th of the original volume. And then he could apply that to his biochemical separative procedures. And so over the course of several months, that's what we did. I just made T cell growth factor, and Rich purified it. And ultimately we had milligrams we had micrograms. We figured we needed 30 micrograms per injection. Now, what am I talking about? We had micrograms. We'll get to milligrams later. So then we also didn't put all our eggs in one basket. We immunized as many mice as we could. then we immunized them repetitively. You know, 2 weeks later we give them a booster dose, and 2 weeks later we give them another booster dose, and so forth. And so this was really a labor of love and also of hope that this would work for us. And at the end of we could bleed the mice and get blood to test for antibodies in the serum of the mice. And we could use our growth factor assay, and we could also use our TCGF binding assay as a competitor for the radiolabeled binding, and we could detect them. The good news was that over time we could detect activity in the serum of these mice that were blocking TCGF IL-2. So we started immunizing these mice, and that was a hairy thing to do, because we take all this work over months and stick it in a tiny little syringe and then strip it into these mice. And it was almost like, you know, it was really a trust in, if there was a God, you know, if we should sacrifice male babies and maybe we would get this whole thing to work. But over time, we got to be very good at making the hybridoma fusions and then sorting them out afterwards. And we used our assays to be able to see whether or not we had anybody activity in our cultures of these hybridomas and so forth. And ultimately, after several failures to detect any hybridomas, we hit pay dirt. And actually, one of the persons who was really important for this whole sequence of events was Maggie Favada. And she was a technician that I hired on maybe a year or two before we started these experiments. And she was really the hope. She was another Brit, like Doreen. And so she had that magical British accent that made her sound more intelligent than us. It was probably true, you know. And so anyway, I taught her all the assays in the laboratory, the cell culture assays, and she was a natural. And so she and I did these hybridoma fusions and then sorted out the cultures afterwards to try to detect whether or not we had any positives. And actually, I was on a little mini sabbatical in Switzerland with Max when Maggie and Rich hit pay dirt, and it worked. So that was good. And so now we finally had antibodies. And now what we had to do was take these hybridoma cultures and grow them up to larger and larger quantities of these hybridomas that were making antibodies, and test the antibodies for their antibody activity to TCGF and so forth. And that all took a while. But once we had 4 different antibodies from 4 different hybridomas. And we then took these antibodies, and there are methods to use the antibodies to purify whatever the antibody binds to. And so we constructed these were solid phase columns. We complexed the antibody to the solid phase material, and then we would drip the TCGF containing conditioned medium, so we could go back to the conditioned medium, and in a single step, without any other steps, so we didn't lose a whole lot in the whole process. Over time, we were able to purify milligrams of TCGF and IL-2. We could then biochemically characterize it, show that it was really only one molecule, and so forth. And then we had enough stuff that we were able to move forward. Then we could go back to the biological experiments to find out, so what does this stuff really do? And because of the fact that we could say that this is pure TCGF, that was it. Now at the same time, we were trying to also we were extracting from these same cells that we were making to make the TCGF, the protein. We were taking those cells and extracting RNA because we were trying to clone the gene, the cDNA, the complementary DNA molecule that would code for the TCGF. And so we were moving along. We were happy. And at the end of '82, we had the antibodies, and things looked good. And we could also we did experiments to determine the amino acid sequence from one of the ends of the protein, and you can't do that if you've got more than one protein. You can only do it, and that's one of the ways that you can prove that you've got only one protein. So we determined the amino acid sequence, but I didn't want to publish that right away because everybody else was trying to clone the gene for IL, the cDNA for IL-2 at the same time. And if they had that amino acid sequence, then they could use that to try to clone the gene, and I didn't want that information out there. So we sat on our data. Meanwhile, we're moving along as fast as we could to try to get the cDNA, and all of a sudden, in March of 1983, there was a paper out in Nature from a Japanese group that that basically said that they had cloned the gene for interleukin-2. I hesitated to use the word interleukin-2 because I hadn't felt that we had really purified it to homogeneity and shown the world and so forth and so on. Well, it turns out if we compared our amino acid sequence from the protein with their amino acid sequence that was predicted from the gene, they were the same. So after that, I figured, okay, well, we've got it. And the thing is, I'd never heard of these people before. This name was Tada Taniguchi, was the chief investigator who had done a postdoc in Switzerland and learned how to do the cDNA cloning, which was another method that was sort of very esoteric at the time. I mean, everybody was trying to do it, but not as many people were successful. And he had postdoced in Zurich, Switzerland, with Charles Weissman, and that lab was the first one to clone genes coding for interferon activity, and they used the bioassay to determine that they had genes that would code for the protein that would have interferon activity. And so Taniguchi used the same technique to clone interleukin-2. So that was a bummer. That was a big bummer. But, you know, like everything else, you can't win them all, I suppose. But it wasn't, and I couldn't figure out how they had really, they had somehow gotten the cells, gotten their hands on the cells to assay for T cell growth factor activity. And we were the only ones really in the world that had it, that had those cells. So they were a critical reagent. And it was only years later when I was called by some lawyers that were involved in a lawsuit against my first graduate student, whose name was Steve Gillis. And it turns out during the discussions on that, in that lawsuit, it came out that Gillis had sold the Japanese the cells that you needed to assay for T cell growth factor. He only got $5,000 for them. What are you going to do? And so some goes up, some goes down in this whole business. Anyway, we were happy because every cloud has a silver lining. So the silver lining here is that we had confirmation that we really had antibodies reactive with IL-2, and we could generate milligram quantities of it, and we could go on further with the next series of experiments. And so the thing, at that point in time, what we wanted to do was to use our antibodies for the purification step. And of the 4 that we originally got, we knew that one would work better than the others from our experiments, but we didn't know why. And so at that time, a postdoc came into the lab by the name of Ralph Budd, and he had been a rheumatology fellow at Dartmouth, and he had applied to come into the lab as a postdoc. And so we put Ralph onto the quest to find out how these different antibodies, because we knew that they were different from our experiments, how they bound IL-2. And he then used a series of experimental techniques that we had already developed and learned from Alan Monk in terms of characterizing the IL-2 binding to its receptor. And there's a series of experiments, and I'll get into a little bit later with that whole assay, because that's another part of the story. But Ralph did a wonderful job, and we found out how these different antibodies bound, and on the basis of the characteristics of their binding, really how rapidly they would bind onto IL-2, or the IL-2 would bind onto the antibody. And then also how slowly, whether they would hold on to it or not. And we also did binding experiments at different temperatures, 37 degrees being the centigrade, being the physiological temperature. And then we also cooled it down to 4 degrees, which is a refrigerator temperature, because everybody knows that slows down intermolecular reactions and so forth. And on the basis of that, we could improve the efficiency of our antibody purification of IL-2. At the same time, the same kinds of data helped Ralph, and so we published that. And as far as I could tell, that was really another landmark paper in our business, because nobody had really ever done that before with cytokines, and how they would bind to antibodies and so forth. And we used those data then, Ralph did, and he did another series of experiments that we published, which was the creation of the first immunoassay for IL-2. Remember, our original assay was a bioassay, which meant that we were using live cells as the indicator of whether or not we had any T-cell growth factor activity. And that's a research lab kind of thing. The immunoassays are using antibodies to bind to the antigen, and you don't need live cells to do that, and therefore it's amenable to assays that can be used in the clinical laboratory and so forth. And so that was also very, very good. And all this was going on from 1982, 83, 84, 85. And at the same time, my wife, Lynn, had decided to start her restaurant, which she named Café La Fraise. So I was really, you can imagine, I mean, we were busy in the lab. And so, but when Lynn came to me, what happened, I can't remember whether I said this before, but what happened was one of her bachelor uncles died and left her some money. And she came out to me. I was sitting on the front porch of our house in Hanover, and she came outside to me and she said, I'm going to start a restaurant. I said, really? I said, well, that's great. I mean, I thought that was a wonderful idea, because then I knew right away that if she started a restaurant, she would be just as busy as me, and we'd both be busy. And up until that time, she was drifting a little bit, sort of bored, and she'd reached her asymptote with regard to the cooking school that she was doing. And so she needed a new challenge. And meanwhile, the boys were, they were becoming junior high school age kids at that stage of the game. So it was a good time. And so she started what she did was she bought the last single home in the Hanover business district, and she renovated that. So it was an old colonial house from 1826, and she renovated that into her restaurant, and she started her classic French menu. And at that time, in the early 80s, Julia Childs had hit it big in the 60s and 70s. And Julia Child really essentially was Lynn's mentor. Lynn did what the young woman who was named Julie did to make Julia and Julia, that movie, where she went through the Julia Child's Mastering the Art of French Cooking, the page 1, and went through all the way, cooked everything in sequence all the way to the end. And then she started her cooking school, and where she taught all that stuff for 6 years. So she had developed her repertoire during that time, and she also knew all these techniques and methods that she had to learn in order to be able to do that. And before all that, as I was doing different kinds of things in the laboratory and so forth, every now and then she would take off for Paris for, a couple of weeks to a month to 6 weeks, and leave me with the kids. Here I am, I'm trying to push back the frontiers of science, and she's leaving me with the kids. But we made out all right. Anyway, when Lynn started the restaurant, it was great. And most of the people in the Hanover area hadn't really tasted classic French cuisine. Or if they did, they didn't get it all that often unless they went to the big city. And so finally, right in the middle of Hanover was this classic French restaurant. And so it became very popular with the Dartmouth faculty, and they would bring all their visiting firemen when they would come into town as an example of the culture that we had in the North Woods there, and so forth. And also celebrities that came through the restaurant were great. Robert Redford came to Lynn's restaurant one time, and Lynn would boast to people that she touched Robert Redford, meaning that she shook his hand, And then another time, Dartmouth was a very, very conservative institution, politically conservative, because many of the graduates of Dartmouth, when it had gone into business, the Tuck School of Business was a big deal at Dartmouth as one of the graduate schools. And so this was really a Republican stronghold. Dartmouth still is. So one time, Gore Vidal, who was very liberal, came because he was getting an award or something like that. And so he was brought to the restaurant by the Dartmouth theater faculty, film faculty or whatever, or literature faculty. And at the same time was William F. Buckley. And so they were really worried that this was going to start an internecine warfare, so that they put one guy in one room and another guy in another room, and so everything was fine. But it was certainly a great time for both of us, actually. And I look back on those years as really the halcyon years of our time in Hanover. So once we'd gotten purified IL-2 and we could do a whole bunch of experiments that we hadn't been able to do before, one of the things we really wanted to know about was, how does the IL-2 molecule interact with the IL-2 receptor? And for that, we realized that we had to determine the structure of IL-2 on the one hand, but we also needed to determine the structure of the interleukin-2 receptor. Doreen Cantrell was really important again in this whole process, because she used to come to me. She would get to the lab before I did in the morning, and she'd sit and wait in my office. And as soon as I walked in the door, then she'd hit me with all these things that she thought about in the last overnight, And we would talk for about an hour and a half, and then she'd finally exhaust her repertoire of questions, and then she'd go off to the lab. And so one day she said, Kendall, I've met this guy down at the VA hospital. There was a VA hospital in White River Junction, Vermont, of all places. It was only 6 miles down the Connecticut River. And so Doreen said, this guy seems to be a very special guy, and he might be able to help us, because Tom Ciardelli was his name. He was a chemist, and he had an interesting pedigree, actually. he'd done his PhD or his undergraduate work in chemistry at the Rensselaer Polytechnic Institute in Troy, New York. And then he went on from there to the University of New Hampshire and did his PhD in organic chemistry. So that's hardcore stuff, you know. Then he went to one of the Max Planck Institutes in Germany, and he learned peptide synthesis. So this guy would, with all of his organic chemistry, he could synthesize peptides, which are composed of chains of amino acids. And there weren't too many other people that could do that. So he was definitely the real deal. And so we started to talk, and he was working on thymic peptides over at down at the VA. And thymic peptides were, you know, they were sort of in the same school as cytokines, lymphodrec, you know, that they had never been purified, but Tom was trying to synthesize peptides that would have thymic hormone activity, and I dissuaded him from doing that, and I started to educate him about interleukin-2 and how wonderful it was, and T-cell growth factor, and what we were doing, and so forth and so on. And to make a long story very short, I recruited Tom to come up and join us in our labs at Dartmouth, which he did over the course of 6 months to a year. And soon after he got there, so he had a whole different cadre of friends and individuals in the chemistry world, really. One of which was a fellow by the name of Fred Cohen. And he was at the University of California in San Francisco, and he had been doing techniques to determine the secondary structure to predict the secondary structure of proteins. That is how the amino acids would sort of wind around one another, because the proteins really round up like a big ball of spaghetti, essentially, if they're round proteins. And how they do that is really, really fundamental to life, because the sequence of amino acids in the protein determine how it winds up. And it always winds up the right way, and it doesn't fail, because the activity of a protein is determined by its three-dimensional structure, not by the primary amino acid sequence. So from gene cloning and gene engineering, you could determine the amino acid sequence, but you didn't know how it rolled up into its final ball of spaghetti. And Fred Cohen had devised ways to try to do that, and he and Tom got together, and it came out with really the first predicted structure of IL-2. And it turns out that he was correct in large part. And they published a paper in Science, and the structure of IL-2 was 4 helical segments. A helice is a spiral of amino acids, proteins, peptides. So it was like this. So that was good, and we had that. And then another person that Tom brought into the group was a fellow by the name of Howard Hawley. And he was from IBM, and he was a computer specialist. And he was using neural networks to predict the secondary and tertiary structure of proteins. And so Howard and Fred and Tom started working on interleukin-2 and to try to get the whole structure. And you know, neural networks are the kinds of things that are the precursor, that they're used now in artificial intelligence, so that the computers can learn. That's what a neural network does. It learns the most probable thing. So from the primary amino acid sequence, you could get the computer to calculate what the most likely structure would be. So to round out that whole IL-2 structure end of things, we also identified a structural biologist. Now, structural biologists are biologists interested in the structure-activity relationships of proteins primarily, but also nucleic acids and large molecules, what they call macromolecules. Because you got to know the whole structure of the whole thing before you can really understand that. When human beings need to see things, if they can't see it, then it's too abstract and they don't know what to do with it. You know, they can it's like cigar waving. They can say anything they want, really. So we identified a fellow by the name of Ian Wilson, and he's also an interesting guy. He's at Scripps Research Foundation and clinic in La Jolla, California. And he was originally had gone to Oxford and then had done his structural biology work at Harvard. And so I figured that this guy would be really good to get, because we needed to know the structure. And I knew other people were trying to do this as well. So we decided to have a Monterey conference meeting and invite Ian so we could tell him all about at length and in depth about where we were with IL-2 and the structure and so forth and so on. And that was a memorable time. I can't remember whether I've told you about the Monterey Center at Dartmouth. The Monterey Center is a conference center that's on Squam Lake. Squam Lake is where they made the movie on Golden Pond with Henry Fonda and Jane Fonda and so forth. And they were making the movie while we were there. We used to go there every 3 or 4 months. And so we were for these long weekend meetings where we would do science nonstop all day long, and then we'd drink beer and so forth late into the evenings. I was always exhausted at the end of these things. But anyway, Ian came and we discussed everything. And then he set off back to California and started experiments to see if he could get crystals of IL-2, because the way the structural biologists work, the sort of the sine qua non of what you do if you're a structural biologist is that you do X-ray crystallography, which means that you have to, if you want to know the structure of a protein, you have to have large quantities of pure protein. You don't want any other molecules in there, and then you have to concentrate it. And you have to think about salt or sugar solution, so that if you and you know, this is the way they get salt from the sea. They put it out on flats, and then the sun, that's the water evaporate, and then they just scrape off the crystals, the salt crystals, and then that's how they made salt. This is what you do in crystallography, but you do it in the lab, and you do it in a little bit more sophisticated way. You put your protein in different kinds of salt solutions and different pHs and acids and base and so forth, and you just empirically determine the best solution for the formation of crystals. And this takes a while. You can't do this. This doesn't just happen overnight. And so Ian set off on that, and we're doing really well for a while. But now, in 1987, there was a paper that came out in Science by a group in Colorado that had reported the first crystal structures of IL-2. And so they published this paper saying the three-dimensional structure of IL-2. And that was another one of those, took the wind out of our sails, you know. It helped Tom, though. Tom Ciardelli was, meanwhile, he was taking the primary amino acid sequence, and he was making mutations in that primary sequence. And then he was using genetic engineering to make mutant proteins that had the mutation in them. And then he was testing them on how they were interacting with whether they had T cell growth factor activity or whether they didn't, and how they interacted with the IL-2 binding assay. Now, and so that was, you know, that was good. And we still didn't have a clue about the IL-2 receptor. But Tom was off and running. He had and he was recruiting postdocs and doing all kinds of experiments. And then about 4 or 5 years later, everybody found out that this structure that they had originally reported was not correct. It had 4 helices, but they had them switched around in terms of how they were embraced one another. And so all of the mutations, it meant that a lot of the mutations that Tom had painstakingly made, you know, it sort of negated all those experiments. That wasn't so good either. Well, during this time, we were doing an awful lot of experiments on interleukin-2 binding to cells that had the receptor. Doreen was doing these experiments. At the same time, she was using flow cytometry, which was a new technique that really replaced the microscope as the primary method to really study immunology. And she was comparing binding of the number of molecules of interleukin-2 that would bind to cells with the number of molecules of radiolabeled anti-IL-2 receptor that would bind to the cells, since we had this antibody that we had identified with Tom Waldman's group. And the problem was that their numbers were grossly different, 10 to 20-fold different. That was sort of strange. We went back over our calculations over and over again, and it was too big of a difference to really explain by just a calculation error. The other thing is that if we took cells that we knew they had IL-2 receptors, and we could measure the number, and then we exposed them to IL-2, the number of antibody binding molecules went sky high, 10 to 20-fold up. Whereas at the same time, the high affinity IL-2 receptor went down. Well, there was something fishy going on there for sure. So we published that in around 1985, I think, but we didn't really have an explanation for that. But it looked as though there had to be another molecule, another chain, protein chain in the receptor that was responsible for in addition to the first one that the antibody bound to. So at this time, you know, our network helped us out again. Frank Rossetti from the NCI lab told us about a cell line that they had that responded to IL-2, but it didn't have any antibody binding sites. So I said, send that thing up here. We need to look at that. So we did that. And we found out right away that this cell line, and one of the things that Frank had told us is this cell seems to be making T cell growth factor. So if that was the case, and if it responded to T cell growth factor, I wondered if that was why it was growing. And that could be an explanation. It was from a gibbon ape that had leukemia. That could be an explanation of leukemia. The cell just makes its own growth factor. That would be good. So we did a series of experiments, and that was exactly the case. What we did was we used our experiments that we'd done with Alan Monk and steroids, because in normal lymphocytes, steroids would block the cells from making IL-2. I figured if the malignant cells also did that, they'd stop growing, which was the case. And then we could add IL-2 back to them, and they would grow again, which was the case. And so we published that. And so that was really exciting, I thought. I mean, because I'd come from hematology and so forth, I figured we were close on the road to the answer to cancer, and in New Hampshire. But you couldn't really make that intellectual leap necessarily, because you could make the argument that this cell grew out of all the leukemia cells that had been placed into culture, because it was selected for to be able to maintain itself by making its own growth factor. And it didn't necessarily mean that the malignancy itself was because of the fact that they made their own growth factor. Now, about this time, so we're in the mid-'80s and so forth and so on, back in 1983, at the International Immunology Congress, there was a Japanese businessman that was very, very nice to me. Took me out to dinner a couple of 3 times, wined and dined me, and I couldn't understand why he was doing that necessarily, but it was certainly, it took me to really good Japanese restaurants, and that was good. His name was Yukio Sugino, and he was an executive in the Takeda chemical industry. And Takeda is a big pharmaceutical chemical conglomerate in Japan. And Sugino was really an interesting guy. He was also a scientist, and he had gone back a couple of times between academia and industry, getting top jobs in industry, and then leaving industry to go back to Kyoto University from whence he came, which was together with Tokyo universities, the best universities in Japan. And then he'd go do that again. He'd go back to Takeda or Kyoto and so forth. And so we had these long discussions. He maintained that the industry was the best place to do science because they had better resources and they also had better people. And I said to myself, well, that may be true in Japan, but in the United States, it ain't true. And the best people are in academia, and we've got the government giving us all the money that we need. Anyway, he contacted me after I had met him in Japan, and said that he wanted me to come and visit them again, which I did, and he introduced me to another fellow by the name of Koichi Kato. And Koichi was an established scientist within the company of Takeda, and Dr. Sugino asked me if perhaps Koichi could come and do a year's sabbatical and come into the laboratory and, learn how American scientists do their deal and so forth. And so I said, sure. So Koichi came, and Koichi was a practiced biochemist. He knew his stuff. He knew what was going on. And he was very, very good. And he was also a very interesting guy. He was a tall Japanese guy. He was much taller than me. And that he was also very, very personable. People liked him immediately. And so he was, I'll never forget, I went to the airport to pick him up, and it was a snowy day. I can't remember what month it was, but it was, this wind was blowing and the snow was coming down. And he had just landed in one of these little 20-seater planes that comes out of Boston. That's the way I got used to get out of Hanover. I could go to Boston, it would take 45 minutes as the crow flies. Or I could go to New York at an hour and 15 minutes, which was both of those were very, very good. And the airline was called Air New England. And the Dartmouth students nicknamed it Air No Engine, because you were sort of taking your life into your hands in this plane. Anyway, he landed, and he came down the ladder, and I can imagine it was a shockaroo, because life in Hanover was not the same thing as life in Japan, which is much more temperate than the north. He came from Hokkaido originally. It's where he grew up, and so he knew how to ski and do all those sorts of things. Anyway, Barb Atherton, who was our administrator, she took him under her wing, and they became fast friends. And she got him and his family situated in across the Connecticut River in Quechee, Vermont. And he did very, very well there. And in the lab, so we decided he decided to tackle the structure, the biochemical structure of the protein that bound the antibody. So he did a series of experiments over several months and showed essentially that that molecule seemed to be present on the cell membrane, both as a singleton and as a duplicate molecule. But there didn't seem to be any other molecule that was bound to that particular molecule, which is what we were really looking for. So at the same time, another Japanese postdoc came about the same time, and his name was Keisuke Teshigawara. And Keisuke was a postdoc. He'd been a student with Junji Yodoi, who we became very friendly with. And Junji was best friends with Takashi Uchiyama, who was the one who had generated the antibody that was now binding to the IL-2 receptor. So it was sort of an incestuous little group of people all over the world that were involved in this whole business. The thing is that Keisuke brought with him, or obtained from Junji, another cell line that was like the gibbon ape cell line that we'd worked with from Frank. This cell line looked as though it was different from the T cells that normal T cells that we were used to working with, because it had an excess number of binding sites that didn't bind the antibody. So we took that cell line and we cloned that whole cell line. So we got all these individual clones, and ultimately what we found was a clone that only had this non-antibody binding IL-2 receptor and that's what we were looking for, because we figured, because the hypothesis was, well, there's another chain. And so Koichi, together with Keisuke, and also a graduate student, Hui-Mei Wang, who was a Taiwanese person who was a Dartmouth graduate student from biochemistry, she came into the lab too. So this became the receptor team. And they all worked on the biochemistry and the structure of the IL-2 receptor. And over the course of several months, we had really unparalleled data that indicated that we had another binding chain, another part of the IL-2 receptor is what we thought. And so we published that, and it came out in January of 1987. In the meantime, two other groups had come out in like November and December in 1986, basically also concluding that there was a second chain to the IL-2 receptor. So that was sort of a landmark period between 1982, when we developed the first antibody, and 1987. 5 years went by. Finally, we know that, okay, that was the second chain. Well, and then, so we took that data and we wondered what was going on. And we had some cell lines that had one chain and another one that had other ones and other ones that had both. So we had all kinds of reagents that nobody really else did in the world. And so Hui-Mei Wang, she decided to do these experiments that Alan Monk had taught us to do in terms of IL-2 binding experiments. And there's two different kinds of IL-2 binding or hormone binding experiments that had been established at the time. One is called an equilibrium method, and the other one is a kinetic method. The equilibrium method, you just mix the cells with radiolabeled hormone and let it come to equilibrium, which doesn't take very long, 5, 10, 15 minutes. And then when it's in steady state, you stop the reaction, and then you can enumerate the number of molecules bound and so forth. But you don't know how fast everything is working. It's not a dynamic, it's a steady state experiment, whereas a dynamic experiment is that you want to know how fast it's going on, and if you wash it off, then you want to know how fast it comes off. And so we had all these cells, different chains and so forth and so on. And so Hui-Mei, for her graduate thesis work, she did a yeoman's job. She just did a beautiful, beautiful job, and showed essentially that the way this receptor was working was both of these chains were binding the molecule, which meant that they had to be binding different areas on the IL-2 molecule. And one chain, the first chain that had been identified, it bound IL-2 very, very fast. I mean, within seconds, it was on there. But it also came off very fast. If you then added the second chain, bound IL-2 much more slowly when it was going on, but it also held on to it much better. The way you make a very high affinity or a very efficient receptor is you grab onto it very fast and then you hold on to your dear life and you don't let it go. That's what high affinity really is, essentially. So we published that also in 1987. And so that was great. And we were, we were rolling. It was fun. So just about this time, I got a letter from the NIH telling me that they had, my time came up for a grant renewal. So I figured this is not a problem. I mean, we got so much data, we don't even know what to do with the data. And I was shocked when I got this letter from the NIH because they said that the grant review committee, the peer review committee, had several questions about my application. And so they wanted to send a site visit team up to Dartmouth to go over everything. And I said to myself, that's a shocker. I mean, they don't site visit individual grants. They routinely would site visit what they called program project grants, which were about 4 or 5 individual grants put together so that 4 or 5 investigators were all going to cooperate on something. And they were big deals and very complicated. And so site visit people would go to the site and they would, of the principal investigator, and they would go over all the experiments. So I said to myself, this is, if they don't fund this, we're going to be dead in the water. That would have been a catastrophe. Because then the money would be no longer, all these good academics, science funded by the government would go away. Anyway, so they sent the site visit team up. They stayed in the Hanover Inn, which was just down the street from my wife's restaurant, Café La Fraise. And the night before the site visit, the administrator of the site visit team and I went out, he took me out to dinner at the Hanover Inn. Should have gone to Café La Fraise, but we went to I figured he's the one. So he wanted to get to know me and so forth. And we had a long dinner. We went over a couple of 3 hours over all kinds of things. And I explained to him all about the science and so forth and so on. And at the end of it, he said, okay, well, listen, he said, what you need, you don't want to be confrontational with the site visitors. And most people who know me know that this is sort of my natural way of being, isn't confrontational. And so he says, you don't want to be confrontational, and you want to be professorial, you want to be charming, you just want to explain everything as best you possibly can. So after a night where I didn't sleep a whole lot, we had this site visit, and we had it in the dean's boardroom, which was all paneled and, terribly old school, very impressive. So we had it in there, and over the course of a whole morning, we had to organize all this. We had to practice it, everybody, because it wasn't just me. It was going to be everybody in the lab was going to stand up and give a little show and tell. So, as I said at the site visit, everybody in the lab participated, and they were all really good, far and above everybody else. Koichi Kato was a star. Because it was clear that he really knew what he was talking about, and he was the real deal, and the data were the real deal, and that really sealed the envelope and made us become refunded, which was a good thing. And so we did all that, and I was professorial, and so as much as I could be. And then for lunch, I had it catered by Lynn's restaurant, and And so it was really good food. So they brought this all into the dean's boardroom. And then after lunch, then everybody was very happy and so forth and so on. And ultimately they when they all left, they gave word back that we had passed in flying colors. And so that was good. And that's the end of this chapter. Thank you. So if you've enjoyed this video, please like, subscribe, and sign up for my newsletter, where I'm serializing my new book, which is called The Quest for New Knowledge. You'll find a sign-up link below. Hey, thanks again. It's been great.